All right, so welcome to today's webinar. My name is Kyle Shavasta, Public Programs Associate at the Community Health Training Alliance. This is our second installment of our webinar series focusing on HIV cure research. Last week we had an HIV research update discussing the work being done at the Beat HIV Delaney Collaboratory, a consortium of more than 60 top HIV researchers from leading academic research institutions working with government, nonprofit organizations, and industry partners to test combinations of several novel immunotherapies under new preclinical research and clinical trials. That webinar was presented by Dr. Luis Montaner, who provided a robust overview of the past, present, and hopes of what HIV cure research could yield. Kenneth Lin, who is the Managing Director of Clinical Studies, was also on hand to provide more perspective on what a trial participant could expect. Today we are happy to welcome Ken back, and equally as enthused to have Bill Freshwater, Beat HIV CAB co-chair, with us to talk deeper about what it is like to be a participant in an HIV cure-directed research trial for the Beat HIV Delaney Collaboratory specifically. They are going to describe why the search for a cure is important even with effective treatment, and explain what potential research volunteers might expect in a cure-focused clinical trial. Now, before I hand it over to Ken and Bill, I'd like to go over a few housekeeping rules. So forgive me while I close this one window out that's causing a technical issue. So every participant or every viewer is on mute. If you have a question, there is a Q&A box at the bottom. So just ask those questions there, and we will field all questions at the end of the webinar. All participants will receive a certificate as well as a survey, just to give us a brief overview of how this went. So thank you for that. And without further ado, I would like to hand it over to Ken and Bill respectively. How are you both doing?
Fabulous, thank you. Good afternoon, everyone.
Good afternoon.
Okay, so this is Ken, and I think I'll take the lead on this and give a brief overview of how we want to proceed this afternoon. So I'm going to initiate the conversation, give an overview of the expectations one could expect from joining or considering a curative research study. And then I will speak projects that are open at a collaboration between the University of Pennsylvania, Philadelphia site, and the Wistar Institute under the umbrella of Beat HIV. I'll then turn it over to Bill, who will offer his perspective, having actually been a participant in a curative HIV study at the NIH, as well as his point of view as the co-chair of the Beat HIV CAB. And the CAB consists of people in the community, either directly or indirectly dealing source to the clinical team and the researchers to ensure that we are focusing on issues that are important to the community and ensure that what we're putting out there will have the support of the community. Because if we come up with something that makes total scientific sense, but it is not relatable to the actual community, it would be a failure. And then finally, after Bill has an opportunity to express his thoughts, we're going to open it up to Q&A, and that'll be an opportunity for folks to type in their questions, and then Kyle will present them. And then between Bill or myself, we will do our best to answer those. So I'm from Philadelphia, born and raised. I was growing up in the gay community here in Philadelphia in the 80s when HIV began to be a topic and took over the lives of many people. I've had friends who have been ill from this disease, and I've lost several friends as well. I was not a nurse at the time, I subsequently became a nurse and have now worked in the area of clinical trials in the area of HIV for the last 12 plus years. So when you're dealing with a clinical trial, there's a lot of activities that happen even before that trial is presented to you, either by an advertisement or a doctor or a friend. There are committees within the government to ensure that the protocol or the plan is designed in a way that is safe for participants and that all issues have been calculated and are safe as best we can, at the same time trying to be cutting edge and make something happen within the care of folks dealing with HIV. But from a patient standpoint, after the introduction, the very first thing they will encounter is an informed consent. An informed consent is a document and a process, which is touching the process from the very beginning all the way through. So once a person gives their consent to participate, it is actually reaffirmed during every encounter that they wish to continue and that they realize that at any time they could change their mind and exit the program early if circumstances have changed in their life. So in the informed consent is the who's, how's, what's, and why's. What procedures will be done? How frequently will they be done? Why do they need to be done? What are the risks of having them done? How much time is going to be asked of you as an individual when you're joining a study? Now, there are varying types of projects, and one of the most complex and time-consuming would be an HIV curative study, because they typically run in about a year's length from the very first contact until the very final follow-up visit. They often include collections of blood, fancy collections of blood where we're focusing on certain cells, rectal biopsies, which is a tiny collection of tissue. Medications are often asked to be taken during this time. So as you could expect, the document is going to be some length to it. So you could expect a document, and don't be shocked if it were 20 plus pages, in order to adequately explain what the project is about, what you're asked about, what safety parameters are put in place, and then a lot of information regarding HIPAA and your privacy is also included in that process. So the document is given to the patient. They go home with it, pick their favorite crayon or marker, and begin to mark up their copy. Circle words that are not clear to you, star areas that are of concern to you. By the time that the document is totally read multiple times by the person, it should be nice and bright colors all over it. You're allowed and encouraged to share your reading of that document with your spouse, husband, wife, partner, someone in your religious community who you value their opinion, family members, other patients. So you're not restricted with that document in any way, and we actually encourage people to share it with others so that you can get as many opinions on your decision as possible. And then finally, you would then bring this document into the hospital and sit with the doctor or the nurse in charge of the project. And then page by page, we would then reconsider the whole protocol, giving you an opportunity to answer any question you have. It's only then, once all those questions have been asked and answered, that we would then ask you, do you wish to proceed? And then by signing on the line and countersigned by the nurse or the doctor is when the first step towards being actually in a clinical trial starts. So I've spent a decent amount of time with that explanation because it is fundamental to having somebody go in with their eyes wide open and truly being informed about what they're agreeing to get into. And again, I would reemphasize that it is a volunteer project and that at any time during the process, the person can change their mind and rescind their consent. There is compensation for being part of studies. They're meant to compensate people for the time and effort and travel expenses needed in order to come and meet with us. I always encourage individuals that if they are purely doing a trial simply for the financial monies, then those are not necessarily the individuals that we want, because they're not truly giving consent. They're focusing too much on what is relatively small amount of money, which is meant to simply make sure you don't have to come out of pocket. And it should be a factor to know that you're not losing money as being able to make these appointments. But if it becomes too much of a motivating factor, then these individuals are not necessarily the ones that we want, because ethically that isn't a true informed consent if they're focusing too much on compensation that they might be direly in need of in their life. Once the consent process is done, then we move into an area called the screening process. This is a visit or visits where we draw blood, we undergo small tests, could be an EKG, blood pressure, weight. And what we're trying to do at this point is ensure that we have individuals that have similarity to them, so that everyone going in the study, for example, they have controlled or no history of diabetes, or their weight has to be within a certain range. These are for both for the scientific aspect, but also for the safety of the individual. We're trying to ensure that they could participate in the trial in a very safe manner. Once we get through this block and that we've seen that they've met all the criteria, we actually go to enrollment. Enrollment involves a lot of collection of information and collection of biological specimens. These are pretty much the before picture of how things are with the individual before they actually start any regimen or new medication that we plan to use as part of the study. These collections could be the traditional blood. They could be an apheresis collection. An apheresis is a fancy form of blood collection utilizing a machine similar to a dialysis machine where blood is filtered. Certain cells are collected into a research collection bag, and then the remainder of the blood is then returned to the patient. This is a very safe technique, and it allows us to focus on the collections of special cells while having no waste, because all of the blood that we were not interested in is returned to the individual. And then finally, another type is, as I alluded to before, rectal biopsies. The rectal area, we usually have to go in 2 inches, and little tiny microbiopsies are taken in about the size of a tip of a pen, the writing point of the pen, very tiny. And what that information allows us to do is measure the reservoir of the individual before anything is done. So that, again, this is the before picture, which allows us to evaluate the effectiveness of any regimen or medical treatment that we plan to use during the trial. We then begin any kind of medicines that are going to be required as part of the study. Not everyone in the study frequently gets the same thing. There's a function called randomization. Randomization is where certain individuals are determined to receive a combination of medicines in group A, while another group may be receiving medications as form of group B. And this is done by computer, so that to avoid any kind of bias on the team who is randomizing, to ensure that all the men are not put in one group or all of a certain race are put into another group. It's truly randomized in order to evaluate the effectiveness of medicines in Group A versus Group B. Typically, there is a loading process where an individual is on their HIV medications and begin to receive these medicines, usually about a month. So that you're actually receiving your traditional medicines as well as your investigational medications at the same time. It's at the end of that 1 month that we then go into an area of research required. It's called an ATI, an analytical treatment interruption. Basically, in order to evaluate how the experimental meds are working, in a cure study, we have to stop the traditional medicines that an individual has taken in order to evaluate that effectiveness. Now, this is often an area that requires a fair amount of discussion with patients because for years and years, we have constantly said, take your meds, take your meds. And in this environment, where you're closely being monitored by doctors and nurses, the only way we can evaluate the effectiveness of a cure strategy is for the individual to stop taking their medicines. Now, during this phase, they are monitored anywhere from once a week or once every 2 weeks, having their viral load and their CD4 and other lab values monitored to ensure that the person maintains safety during this time. Once an individual's numbers start to skew out of that safety zone, which is already determined as part of the protocol before we even start, once they have identified that and gone into that range for 2 consecutive tests, they then revert back to their HIV medications that they started with. Now, the consent will say there is a small opportunity where an individual will return to their medicines and they will not be effective. This is true from a scientific standpoint, but it has been our experience with the 80 to 100 people who have undergone ATIs with us in the past, that everyone who we've worked with has been, in fact, able to return to their same medication regimen that they started before the study took place. So there is a small scientific chance that someone may get resistant. However, when they follow the directions of the study team, we have not seen that. If that were to occur, there would be genetic testing done, and we would identify a replacement HIV regimen for them to go on to. But knock on wood, to this date, we have not had to exercise that procedure. It's the ATI that gives us the information of how effective we are with the experimental medication regimen controlling the HIV in absence of daily medications. The goal of any curative study is the reservoir, the hidden pockets of HIV that we measured during the early enrollment phase. After the experimental medications are used, we measure it again. And the goal is to show that there was a decline in that reservoir. And then in many trials, they'll even have a third collection 6 months after the individual has already returned back to their normal HIV medications to then look for what happens 6 months after everything has normalized. So then they would have a collection at the beginning, a collection immediately following the HIV experimental medications, and then a third some 6 months after the person had returned to their traditional HIV daily medications to see what kind, if any, of a long-term impact that the research trial medications had on these reservoirs. It is these reservoirs that prevent a cure for HIV. If an individual were to stop their medicines, their traditional medicines, these reservoirs would wake up, move back into the blood, repopulate, and start the whole process all over. So the heart of any cure project is going to be coming up with a strategy to attack and eliminate those pockets of reservoir. And so that is true for any HIV cure trial as well as ours as well. And then finally, there's a follow-up phase. So now the individual has done all the aspects of the study. They've returned back to their medications, and then we follow them for usually a minimum of 3 months with frequent check-in visits to ensure that all their lab values and any kind of side effects that may have encountered during the actual study have been resolved. So the real cut and dry approach would be is we want to put the individual back to where we found them when they initially started the trial. So we don't want to have any kind of a problem after the study that the individual didn't have coming into us. So patient safety, as you can see throughout the entire process, whether we're seeing them weekly or we're monitoring their labs to ensure that their viral load and CD4 are being safely maintained, and then even into the follow-up phase, we're looking again at safety to ensure that they have returned back to their pre-study numbers and situations. So the consent process starts. So a 2-week process. So this is not a agreement to enter in lightly. The individual is looking at multiple procedures and multiple visits and blood draws. However, they are incorporated into the process and are kept aware, as is their primary HIV doctor throughout the process. Just because an individual starts a research project, it does not mean that they are no longer under the care of their original doctor. Their doctor is actually collaborating with the team. Anything that's going on within the procedures and blood tests is being shared with the individual's primary HIV provider so that they know what is going on and can actually communicate with the team if there's any changes in an individual's care. That's also important. A lot of times people are concerned that they've been seeing the same doctor for 10 years and now they're coming in and putting their safety and life in the hands of people who they've only recently met. And so that is a big important thing to recognize, is that we are not asking them to leave the care of their initial doctor, but rather expand it with the addition of new folks. So that is an overview in a generic sense of cure strategies. Now, Kyle, if you could advance to the next screen, there's a graphic here that kind of lets us know what is going on in the Beat HIV collaboratory. As Kyle referenced earlier, we are a team of 60 plus with ties around the world, while the clinical activities are happening here in Philadelphia, which is my area of expertise. The actual specimens and work is split around labs in Europe, throughout the United States. So the first goal of any of the Beat HIV collaboratory is goal 1 is to find out where and how HIV hides. I referenced the repository, and that is basically it. Is when the virus is being controlled in the blood by our HIV medications, some of the HIV goes dormant and goes into these reservoirs deep into the body. And you can almost imagine them putting a hat and a wig and glasses and a fake nose on and mixing with all the other healthy cells of CD4s and waiting for the day that they can rip off that disguise and re-enter the bloodstream. And so we're trying to get better and better at finding these cells. Often we're looking for a cell, literally 1 in a million, that we're trying to get the immune system to find it and kill it so that it won't be a problem for us in the future when the individual were to stop their medications. I referenced the intestinal tract when we were doing our rectal biopsies. The intestinal tract is a favorite place for the immune system to relax during its off time. I often describe it as like the Las Vegas Strip of the body, where the CD4 cells go and sit by the pool and have a drink. And so wherever there's a lot of CD4, there's going to be also HIV. So that is an area why we collect specimens from that area, because it's relatively easy to get to within 6 inches of insertion. And we can get a sense of what's going on with the stored dormant virus before and after an HIV medication during a clinical trial. Now, specifically for Beat HIV-2, this is actually our 5th study using interferon as at least one of the mechanisms to try to get the immune system to go after these hidden pockets of virus, the reservoir. Now, in Beat HIV-2, we're actually, as you can see on the weights of this graphic, interferon is on the one side, and the abbreviation AB is on the other. That is antibodies. There's another group of researchers that are part of our team in Rockefeller University who have used as a way of going after the reservoir. And so we're combining these two strategies in Beat HIV-2, familiar with, and the use of these antibody infusions, bringing the two of them together and see if there's any synergistic benefit to them two being used together within the regimen as far as reducing the reservoir. And so this is the first time that we've worked with antibodies. We're excited for it. Antibodies are typically made by the body in response to an antigen. That could happen through exposure to a bacteria or virus, or it can happen through inoculation or a vaccine. Now, we have been unsuccessful in creating an HIV vaccine. However, Rockefeller was successful in creating 2 antibodies in the laboratory and then given to an individual during these research projects. The antibody's life cycle is about 3 weeks. So then we would then have to reinfuse them every 3 weeks during the trial. In our trial, it will be a total of 7 infusions, 3 weeks apart from each other. And it just is a matter of sitting in a chair and having an IV started, and then these antibodies infused separately. It takes about 2 hours per antibody, and it gives us a resource in our immune system to potentially go after these hidden reservoirs. So we're excited to see what the impact of the antibodies when added to interferon does. Now, we also have another trial opening up. It's more a gene trial, gene modification trial, where we actually remove some of an individual's CD4 cells. We modify them. In this graphic, we show that we arm it by giving it a sword, giving it a weapon that it didn't have before. And we give it the shield, which is we give it the ability to resist HIV infections in a way that it didn't have before. And then we give those newly superpowered CD4 cells back to the individual and see how those cells fare fighting the HIV reservoir. So these are the 2 trials that are up and going. We are currently pre-screening for Beat HIV-2. By that, I mean that we're checking to see whether antibodies are, if an individual is sensitive to the antibodies we plan to use. So if that is the case and the individual is sensitive, then they can continue to be considered for the underlying study. So we are actively speaking with people and having a small blood draw appointment, which is all being done with me. Kyle, advance slide, please. The next slide will show my contact information. If you or someone who you care for, whether it's a case manager or a physician or a social worker, anyone can call me directly. Kyle, can you advance the next slide for me, pal? Great. And so there's my contact information. You can contact me directly by phone or email with interest or questions, whether you want to be pre-screened or someone actually just has questions that are more conducive with a one-on-one conversation. I'm always happy to speak with individuals regarding our work. Not everybody is going to be an ideal person for a research study, and that's okay, because we have to pick people who are similar to each other, and we have to pick people who are safe to be able to do a trial like this. But even if you're not eligible to actively participate, there are ways to support our research. You could be a community ambassador. And what I mean by that is that after they're done speaking with me, they now know a decent amount about the trial. Just simply mention it to other people in the community and motivate them to call me, because it's the word of mouth, it's community support that sometimes gets me the people that we need in order to participate. We also have a community advisory board. These are the folks that are advising both the clinical medical aspect of the trial as well as the researchers on how best to present the trial and factor it that's best for the community. So a community advisory board, or the CAB, is a group of people who are impacted either directly or indirectly by HIV, and they offer feedback to help make the outreach much more effective to the community. A contact person for that, if you have any interest in participation in the CAB, would be Beth Peterson, and Beth can be reached at 215-898-3934. Beth Peterson is the Beat HIV Program Manager at the Wistar Institute, and again, her number is 215-898-3934, and Beth could give you information about how to potentially become involved in the CAB. And then finally, one last number I'm going to give you is if you're a patient at Philadelphia Fight and you have interest in the curative study Beat HIV-2, your contact person there would be Lyndon, L-I-N-D-E-N. Lyndon is the research program manager at Fight, and her direct number is 215-525-8695. 215-525-8695. And Lyndon would handle any direct inquiries from Philadelphia Fight patients. Any other patients or people from Penn or any other clinic throughout the city would first reach out to me at the number on the screen, 215-662-8217. So at this point, I've given my spiel on my aspect of it. I'm going to turn it over to Bill Freshwater, who will give his comments with regards to cure HIV research as both a participant and as a member of our CAB. And then finally, we'll open it up to questions. So I'll turn it over to you, Bill.
Hi, thanks a lot, Ken. Good afternoon. My name is Bill Freshwater, and I'm really happy to have the opportunity to speak with you today about my experiences with cure studies. To start off, I'm a 62-year-old HIV-positive gay man. I'm not a doctor or a scientist. I am a member of the Philadelphia Beat HIV Community Advisory Board. One of the missions of our Community Advisory Board is to integrate community involvement in HIV and AIDS cure-related research and clinical trials. That's one of the reasons I'm here to talk to you. But in addition, back in the early 1990s, I was on an HIV vaccine trial, the Salk Immune vaccine study. And then during 2017 and part of 2018, I was on an ATI, or Analytical Treatment Interruption Study at the NIH. I think Ken mentioned that. My participation in these two very different types of studies done at very different times in the AIDS epidemic at different times in my life helped me to be able to share with you some reasons why cure studies are vital and some things that anybody contemplating participation in a cure study should consider. I'd like to start off by reminding everyone of the trajectory of the AIDS crisis. I know, this may be old hat to some of us, but to many people, it's a thing that happened so long ago that the raw pain and fear that we were facing is not really imaginable. Back then, a diagnosis of HIV was a death sentence. There was no treatment. People didn't want to touch you, and some doctors and dentists wouldn't even treat you. For me, back then, people jumped at the chance to be on the Salk vaccine study. I'm sure that there was some form of informed consent, but I doubt anybody looked at it. I know I don't even remember it. Because this study and AZT were basically your only options. And then in about 1995, the first class of HIV drugs were introduced. These were protease inhibitors, and they were a real game changer. Looking back, it seems like overnight, your friends and lovers stopped dying. I know it's strange to use the word lucky when talking about having HIV, but those people, like me, who were able to hang on until these miracle drugs were available, had a chance, a shot at life. And this chance was due to the brave and selfless people who took part in clinical studies that helped develop these drugs. The desire to acknowledge and repay those people is one of the reasons I decided to take back in a treatment interruption cure study. But before you can consider taking part in a study, you have to hear about the study. You have to learn that a study is being conducted and what it's all about and where it is and who can participate. I learned about the study I joined by talking to a friend who does HIV research here in Philadelphia. You can also find out about studies at your doctor's office, at community centers, or health clinics. Once you find out about a study that sounds interesting, you really need to think about all the ways this could impact you. A great way is to start to discuss the study with people, a lot of people. I started my discussions with my husband. I'm fortunate here because my husband is a physician. He's not an HIV doc, he's more of a scientific research type. But he was able to help me read and try and completely understand the informed consent, which explains the study in detail. Next, I made an appointment with my longtime HIV doctor to discuss the possibility of me joining the study. He had been my doctor for about 13 years, and at first, he was reluctant for me to participate. He explained to me that his primary concern is my health. Any study, including an analytical treatment interruption study, has risks. As he put it, why rock the boat? I understand that concern. I share that concern. My husband had the same concern. But I spoke at length with the study doctor and the study coordinator. And believe me, there's no question that can be considered too stupid to ask. I firmly believe you must be your own advocate. And part of what we are doing as a community advisory board is to try and make these informed consent documents easier to read and to understand for the participants. Like Ken said, any word you don't fully understand, you need to get defined. This is not the time to be shy. Then, after talking with my medical doctor, I had discussions with my psychologist about this. You've got to really think about the psychological impact of the study and not just the physical impact. What I would have loved would have been able to discuss what a study is like with somebody who had already been in a study. And that's why I'm really excited to take part in this webinar. I think that to hear from a past study participant is different from hearing from a doctor or a study coordinator. Then, in order to have meaningful discussions, I had to identify what I considered to be the key issues that required extra consideration before deciding to join the study. And there are both pluses and minuses to consider. The plus side includes the altruistic reasons I already mentioned, but there's also a selfish reason. Maybe the new therapy you're going to get is the cure. And if so, being on that study will put me at the head of the line to get that cure. And that's a good thing. You can't overlook the increased care you receive on a study. I'm a Medicare patient. My quarterly doctor visits last about 15 minutes, and then I get my lab work. On a study, you get way more medical attention. I got a complete physical every visit. You do have to remember that when the study's over, you go back to your regular doctor visits, so enjoy it while it lasts. And as Ken said, make sure that all your lab results during the study get sent to your regular doctor, because that's who you're going back to. On the minus side, a study has risks. My main concerns were about the stopping of my medications or the treatment interruption. I wanted to know how many studies had been done that included a treatment interruption. Had anybody died from a treatment interruption? When would I stop my meds? When would I go back on my meds? Will I be able to go back on the same meds I was using? How can they be sure I won't develop resistance to my drug regimen? Why do I have to stop taking meds? I mean, I was much more concerned with these things than I was about the experimental drug I would receive during the study. Everyone has to decide what are the key issues for them and then get comfortable with the results and the responses to these issues. And don't forget to verify that the study you're considering is being conducted by a reputable organization with certified medical staff. There's a website, clinicaltrials.gov, to verify your study. You also need to be clear about any possible costs associated with the study. Will your insurance company be billed for the usual standard of care type of lab work that you usually get done? And if so, you've got to verify that the study's medical provider is in your insurance network. Will you be reimbursed for expenses associated with the study, like transportation or lodging? As a matter of fact, on my study, I was getting $120 for a night for a hotel lodging in DC. And let me tell you, trying to find a hotel in DC during cherry blossom time for 120 bucks is impossible. So I went to my study coordinator and said, you have to up the ante. I mean, I can't pay $250 and only get $120 reimbursement. And I think, on a study, as I say, you've got to be your advocate. You've got to speak up if something is not right. As difficult as it may be, have been for me to read and understand and inform consent, think about a potential participant who does not speak English or whose native language is not English. And it's not just the informed consent that they need to be concerned with. What about all the lab tests and exams that you go through? Are translators going to be available? Don't forget to ask for what you as a participant need. Then, once you're satisfied that the study meets your concerns, you need to think about how you'll be able to merge the study with your life. On my study, which lasted over a year, I had appointments basically every 2 weeks. Some of the appointments were relatively short, but others lasted as long as 6 hours when I was getting apheresis, for example. Some of them took 2 consecutive days. This is a very big time commitment. You really need to carefully read and review the informed consent to get a handle on the time that's going to be asked of you. I'm happy to report that some informed consents include a graph or chart that it really shows you how many appointments you're going to have and when they're going to be. Make sure you understand this. Think about how this could impact you, your spouse or partner, your family, your child care responsibilities, even your regular job. Remember that although you can withdraw from a study at any time for any reason, you need to ask yourself upfront if you can deal with the time commitment. Remember, if you withdraw from a study, your slot cannot be refilled by another person. So be sure about everything before you start. After I was able to work through these issues, I made the decision to enroll in my study. At that point, I had to be screened to verify that I met the requirements for the study. Up to that time, I was only thinking about if the study was right for me. Now I found out the study doctors also wanted to be sure I was right for their study. The informed consent sent out the requirements for enrollment. Basically, after verifying your HIV status, they want to make sure you're healthy. And this is for your protection. The study got my medical records from my regular doctor and did some additional tests to establish my base condition. For 6 months on my study, I had biweekly appointments with labs and exams. I also received periodic infusions of an experimental drug therapy. All this time, I continued to take my regular HIV medication. After 6 months, I had to stop taking my regular HIV meds. This would allow the study doctors to determine if the experimental drug could continue to suppress my virus without the benefit of HIV medication. This was the exciting part for me. Could this really work? Prior to enrolling on the study, I was managing my HIV. I was undetectable. I was on a drug regimen I could tolerate. I have access to good medical care. I followed the standard of care, meaning I took my pills. I never missed a dose. I tried to take them at the same time every day. For those of us who have been positive for a long time, thinking about stopping meds is interesting and fun and hard and scary. On the one hand, I can tolerate my meds, and for me, being compliant was not a problem. On the other hand, having a drug holiday would be nice. To not have to think about meds every single day would be great. I mean, the pills kind of take over your life. Do I have enough? Do I need to order? Do I have a valid prescription? You know, if this drug holiday is done as part of a clinical study with medical supervision, that's all the better. Part of the informed consent explains the timing and set points for my treatment interruption. I knew up front what level of viral rebound would trigger the need for me to restart my medications. In other words, how high of a detectable viral load would cause me to have to start taking HIV meds again. I also knew before I began the interruption that I was not resistant to my meds and that I would be able to resume the same regimen. The study had done a genetic test to identify any potential drug resistance. My study said that if after treatment interruption, I had a viral load in excess of 1,000 for 4 consecutive weeks, I would have to restart my meds. It's important to note that this viral load level is low and the time period is short, and that's for the health and protection of the participant. In any study, the biggest concern should always be the health and well-being, both physical and mental, of the participant. Of course, stopping meds means that you have a risk of becoming viremic and thus being able to transmit HIV. This was discussed at every visit. Condoms were available. Safe sex was discussed. For a long-term survivor, this is not new ground, so for me, I did not think it would be an issue. But I have to say, during my treatment interruption and between visits, I fell and cut my arm and bled heavily. It's no big deal, except I was with 2 of my young cousins, and they immediately wanted to jump in and help me. And I tell you, all the old fears of blood transmission came back. I have tainted blood. That was in my head. I hadn't thought about that for years because, now it's U equals U. Undetectable equals untransmittable. But with a treatment interruption, you're not sure if you are still undetectable. That started me thinking about the time between blood work and the time to get my results from the visits. That's why I feel strongly that we should all be advocating for people to work on a home test kit for viral load. All it has to do is say whether you're detectable or undetectable. That would go a long way to relieving stress in a study. For me, my treatment interruption lasted about 2 months. At that point, I had to go back on meds based on the study criteria. It's funny, but I asked if I could go longer and see if my viral load would drop back to under 1,000. I mean, I knew it wasn't really a possibility. You have to adhere to the study criteria. But I just wanted to have a little while longer to hope that maybe this was the cure. Maybe I would not have to take meds again. I'm happy to say that within 6 weeks of restarting my medications, my viral load was once again undetectable, and it has remained undetectable ever since. I need to stress that I had no safety concerns during the study. I saw more nurses and doctors and had more blood tests and physicals than I ever had had. I knew who was conducting my study. In fact, you get kind of an ID card from the study coordinator with phone numbers on it in case of emergencies. At one point, I got called for jury duty during my study, and explaining to a judge that you're scheduled to receive an experimental HIV drug infusion and that the timing cannot be changed is a very good reason to get out of jury duty. My privacy was protected throughout the study. All the vials of blood collected during the study used a study participant number, not my name. My study coordinator called me with all my results. You also had the option of getting all the results through a patient portal on your computer. And I have to say, I really like that my coordinator called me before every visit to remind me of an upcoming visit. My study was ultimately not successful in that the experimental drug did not suppress my virus. But me saying that my study was not successful is wrong. I knew going into this study that it may not be the cure. Every study is a step on a path to the cure, and every step is important. Remember, if no one participates in a study, there will never be a cure. The lifespan today for someone diagnosed with HIV is vastly longer than it was 25 years ago. That is due to clinical research and studies that involve human participation. Just because we have easy, readily available one-pill-a-day treatments for some doesn't mean we can be complacent. We need to continue to search for longer-lasting antiretroviral treatments. We need to have cure studies that include treatment interruption. We must continue to move forward. Thank you.
Thank you, both of you, for that wonderful presentation. So it looks like we actually do not have any questions from the participants, but I have a few questions, if that's okay.
Sure.
This one question is for both of you. There's something very outward-facing about this study. We have conducted these webinars. There are going to be CAB members at the Prevention Summit on June 11th at the PA Convention Center to talk about various topics, including their participation in the study. And there's going to be a film premiere as well, kind of showing a film of what this study is entailing and just kind of giving a little cinematic scope and perspective to what's going on in Philadelphia. What would you both say is the benefit of having a study like this be so outward facing? Bill or?
So this is Bill.
Yes.
Okay. Go ahead, Bill.
So this is the, so for me, I think it's really important because, as I touched on, I think that some people sort of forget about the need for ongoing research. I mean, we get bombarded, I think, when you look at magazines and you see the, and now it's on TV, of healthy people or healthy looking people in Speedos on a beach, taking their HIV medications. We got complacent. So we need to go out and say, hey, we need a cure, because although we have here in the United States the ability to have access to one pill a day regimens, in many countries that's not an availability. And even here in the United States, in the prison system, you might not have access to pills every day. So the need for a cure, the need to make people aware of the cure, and to the need for people to participate in a cure is extremely important. And that's why we're trying to really reach out and remind people AIDS isn't gone, HIV isn't over, and we need to continue working on this.
I agree with what Bill just tossed out there, and I would also just add, people want transparency, and so they want to know what's being done, how they can participate, and without community support, we all recognize that any effort such as the curative HIV trial is doomed for failure. Mm-hmm. Because Without the participants, we can have the best doctors and nurses and facilities and researchers and money. But without the participants putting their trust and feeling okay to deal with us and to participate, then it's all for naught. Because an individual is buying into the project, but they're also buying into the individuals who they'll be dealing with through that project. And they need to start that relationship from the first introduction. And it takes a lot to build that rapport, and it can be lost very easily. So I think we're just all very cognitive in this world that that outreach and forward looking is the right approach to start off on the right foot.
Wonderful, thank you. Yeah, I definitely believe that transparency kind of fosters community legitimacy. So that's wonderful. That's wonderful. Ken, what would you say to someone who is an ideal candidate, but they're on the fence? They can't quite decide whether or not they want to participate.
I think that if an individual cannot 100% fall on the side of wanting to participate, then I would caution them to just continue to think and pray about it. Because as much as I would want them to join, we have limited spots. In this upcoming trial, there are 21 spots. And as both Bill and I referenced, an individual is reconsenting throughout the process, and they can leave at any time. Now, the flip side of that is that once they take one of those valuable slots, if they in fact exercise that right, then that is a lost opportunity. And so if someone cannot go in, then they really need to really reflect on what's holding them back from that. And if they can't fully commit, then I would say better for them to support the project as a community advisory board member or as a community ambassador and speak to others about it, rather than go in and not 100% be able to buy into it, because I think there's a high chance that they may not be able to see it all the way through. And then that would be their right, but it would be at the cost of the overall project and the community at large. That would be my recommendation.
That's great. Thank you. Thank you. And kind of dovetailing off of that for you, Bill, this being your second time entering a trial, was there any moments of hesitation? I know you said there was, you alluded to the fact that you jumped into it, but was there a point where you put deep thought to it and said to yourself, maybe not this time?
Oh, sure. You really have to think long and hard about this. I feel very strongly about the importance of the need for studies and the need for participation in studies. And for the study that I went on at the NIH, I have received a lot of good feedback from the people I talked to, that it was a good thing to do. And as I alluded to, I think that going on a study is always a personal decision. And what Ken was just talking about, we can't coerce people. I mean, if somebody's on the fence, you can't coerce them to go onto a study.
Absolutely.
It's an individual thing. And I just feel so strongly and so thankful of people who have been on studies that I wanted to do my part and go on a study. And again, there's always the hope that you're going to be on the study that is the cure, and then you're at the head of the line, and good for you. So it was not after a lot of thought, it was not a hard decision. I think it was the right decision for me. Listen, I'm 62 years old. I'm retired. I've got the time to do it. And I wanted to do it.
Wonderful, wonderful. Thank you so much to both of you. Do you have any parting thoughts before we depart today?
I would just like to take the opportunity to just put a few numbers out there one time again. The one thing would be, if you want additional information, please visit our website at beat-hiv.org. That will give you a huge amount of information. What is our current projects, information on past projects, where we see the future going, as well as pictures and bios of all the different people that are supporting the effort, from the researchers to the clinical team to the community advisory board members. It's just full of information. And then again, if anyone on the call either supports or is a patient at Philadelphia Fight, Linden, L-I-N-D-E-N, is the research program manager at Philadelphia Fight, and she would be your first point of contact, 215-525-8695. My phone number, 215-662-8217. I'm happy to speak with anyone, but particularly patients at Penn and any other clinical sites, but I love to talk and be willing to speak with anyone. And then finally, if somebody has an interest in joining our CAB, Community Advisory Board, a point person for that information would be Beth Peterson, P-E-T-E-R-S-O-N, and her phone number is 215-898-3934. And on behalf of Bill and I know that we both appreciate everyone being on the call today, especially as beautiful as it is out there today, leading up to the Memorial Day holiday. We just thank everyone for their interest, and please pass on this information, pass on the existence of Beat HIV, and share all these numbers that I just offered to you freely and frequently.
Wonderful, wonderful. Thank you to you both. And I hope everyone has a wonderful Memorial Day weekend. Take care.
Okay, great. Bye. Take care.
Thanks.